babies lab. Today I'm going to be showing you some of the research that I did for my undergrad thesis. So, looking at a little bit about the background going into this research. When we simulate predatory interactions between species, record their behavior, it lets us make informed inferences regarding their natural predator-prey dynamics, especially in species like bats, where much of their interaction with predators is within roosts and very difficult to directly observe. Mechanisms like threat detection and avoidance enable prey species to survive potential predation. Things like pheromones, in this case, chemicals inadvertently released by a predator, which a prey species may detect and take advantage of. Or visual cues. I see a predator, I avoid a predator. The two species of interest that I looked at for this study were the eastern small-footed bat and the Arboreal roosting little brown bat. I know we've talked a little bit about the rock roosting habits, but primarily in the literature they're considered either building roosters or arboreal roosters, and the eastern small-footed bat being rock roosting. And this key distinction is what I focused on in my research. I looked at two key questions in this research. One, do rock roosting bats exhibit bolder behaviors than arboreal roosting bats when in direct contact with a potential predator? And two, are rock roosting bats more adapted at avoiding terrestrial predator scent cues than arboreal roosting bats? I decided to choose the eastern fox snake as my model predator. One, because they're a known roost predator of bat species, of my own species. And two, because of some other research going on in my lab, I had access to a full shed very easily. Looking a little bit at the methods of the study, I trapped and performed my experiments at 3 known swarming sites around Ottawa, using hard trap as a method of capture. And you can see from my kind of janky setup on the right there, I performed video trials using 6-arm radio mazes, using chase trials as one, and scent trials as another. The video trials were performed as close to capture time as possible, lasted a maximum of 2 minutes, and the bats were released following our data collection, so the same night. More specifically, looking at the chase trials, you can see from my beautiful diagram, the 6-arm radio mazes were modified to be a single hallway with a paintbrush acting as our model predator, sticking through one side of the maze. And this was designed to evaluate whether rock roosting bats are bolder when in direct contact with a potential predator. So first, the bat was placed facing the paintbrush as close as we could get to the paintbrush. It was chased down the maze, all the way down. The paintbrush was brought back to the entrance, and the bat was allowed to reapproach as long as it took, and this was called latency time. And we performed this chase 3 times, or up until, like I said, the trial reached 2 minutes. And then, using my analysis, I compared the time taken to reapproach the predator, latency time, or we also compared behavioral counts for a number of behaviors. This is very simplified, but we have written out a standard procedure to follow for each trial, so each trial is subjected to the same thing. Looking at some of the behaviors that I scored, I find a number of behaviors that were deemed examples of bold, aggressive, shy, or avoidant behaviors. Here I'm demonstrating 2 examples of bold, aggressive behavior, with videos taken directly from my trials. Don't know how well they're showing up if they're a little small, but you can see on the top we have SWAT attempts, which is indicated by a punch or a bat with the arm of the brush, or a bite attempt, which I consider the lunge of the face towards the predator, whether or not actual contact is made with the brush. The methods for these scent trials, another beautiful diagram. The second experiment was designed to evaluate my question of whether rock roosting bats are more adept at avoiding terrestrial predator scent cues. The green circles indicate where the scent cue is placed. The blue arms are indicating the scented arms. The orange arms are indicating the unscented regions, and then the gray area in the middle is what we considered a neutral area, or a scent neutral area. The scent cue was placed in 3 alternating arms. The bat was placed in the center of the maze. Plexiglass roof was placed over top. The bat was allowed to explore for 2 minutes, and then was released following. And then I compared the time spent in scented versus unscented arms to what is predicted by random chance, using a series of parity tests. I performed 2 versions of this trial. One was using predator cues, or the state shed for the eastern fox snake. And what I expected is for them to avoid it. Spend more time in the unscented arms than was expected by random chance. And 2, I used a conspecific cue. I put guano in 3 alternating arms, acting as what I consider to be a neutral scent, and expected the bats to be indifferent to it, spend as much time in each arm as expected by random chance. So looking at the results, the first question of whether rock roosting bats exhibit older behaviors. I performed a mixed effects model using latency time, which I defined earlier as my response, and predictor being species, with a random effect of trial lightning. And what I found is that there was no difference in boldness between species. And though it may look like it in the graph, the appearance of this decreasing latency time, it may be an artifact of there being less time in the overall 2 minutes in the overall experiment, leaving less time for later latency times. However, I am in the process of doing the analysis to see whether there is actual significance there, whether later chase events resulted in shorter latency times, learned behavior. Because I had the data available to me, I performed basically the same mixed effects model, replacing species with sex. Similarly, no difference in boldness between sexes, but interesting nonetheless. Looking at the behavioral counts, you can see on the right there, this is kind of what the chase trials look like a lot of the time. Bats getting scared of the paintbrush and running away. I performed a binary logistic regression, which was a kind of cool way of doing it. I set the eastern small-footed bat as ones with the little brown bats being zeros. And using counts of the behaviors from my defined behaviors, I used those as predictors and saw whether or not any of these behaviors are more associated with one species or the other. The total behaviors that I had were, like I mentioned, bite attempts and swat attempts. I had bouts of defensive posturing. I had vocalizations at a frequency too low to be considered echolocation, or audible vocalizations through the videos, and then non-aggressive contacts, which I defined as direct contact made with the brush, with no attempt to bite, swat, or defensively posture. The results of this were very interesting. We only found significance in that the non-aggressive contacts were more common in eastern small-footed bats than little brown bats. And what I noticed while scoring these videos is that many of the non-aggressive contacts came following later chases in the eastern small-footed bat trials. And this could indicate that small-footed bats have a better ability to quickly realize that a simulated predator wasn't a threat. Quicker to learn, maybe a little smarter than the little brown bats in some cases. But yeah, very interesting. Looking at the second set of results here for the scent trials. Little hard to tell what's going on, there's a lot going on in this graph. But on the left, you can see the eastern small-footed bat trials with the conspecific cue versus the predator cue. Similarly on the right with the little brown bats. And what I found is that neither eastern small-footed bat nor little brown bat spent significantly more time in scented or unscented arms, as expected by random chance. And as I mentioned earlier, I compared the time spent between these arms against what is expected by random chance using a series of predictive tests. Interestingly, however, while the analysis indicated non-significance, maybe because of the small sample size that I had, limited swarming time, lots to do, there does seem to be a trend away from the scented arms in the predator cue trials. The orange arm being slightly higher in the graphs than the blue arms. And similarly, in the conspecific cues, indicated non-significance. However, there is a short trend showing preference towards arms with conspecific cues. So this is something I'd love to evaluate further with the larger sample size. Maybe it could reveal something in the future. So what do I think about these results? Subtle differences in the behavioral scores between eastern small-footed bats and little brown bats could be caused by common contact with small snakes and rock roosts. Lots of non-aggressive contact between the simulated predator and the bats could be a result of desensitization to rock-dwelling snakes that enter roosts but are too small to be considered dangerous. This is sort of what I was getting at earlier when I mentioned that eastern small-footed bats would engage in non-aggressive contacts more frequently in later chases during the trials. Finally, I would love to further investigate the draw to conspecific cues and the aversion to predator cues that weren't supported by the pair t-test with a larger sample size, and see whether those trends actually continue to showcase and start to arise a little further. Thank you so much for listening. I had a blast doing this research and this presentation. Any questions?
So you obviously had the scented and the paintbrush trials. If you would see a stronger effect if you were using the paintbrush smell like a snake?
So that's a great question. It's he asked whether or not we would see maybe see more amplified results with a paintbrush that was maybe scented like a snake. And this is something that I had considered when making the protocol. However, we are limited by animal care protocols. And when you put snake scent on the paintbrush, it's very scary to do that. What I do think, though, is that you would see more significant results. Going back to the scent trials, if there is that slight aversion to predator scent cues, if you do see it at a higher sample size, maybe you would see greater defense, maybe greater avoidance to the predator in those trials. So that's something that I would love to look at also. Yes, sir.
You can,
Okay.
What would make you think that they would find conspecific poop attractive?
Yeah.
Because, I mean, they drop it. They're not normally down mixed in with it unless, I guess, they're crawling on the ground.
Yeah, so the reason that I think that there might be a draw towards the conspecific cues is that it's because of the context. The context being you're being chased in a very confined space by a predator. And I would think if you are using all your senses, your echolocation, you can't find a physical way out. Looking for anything familiar, bats are very social animals. If you smell your friend's poop, maybe that means it's safe over there. Yeah, there wasn't too much I could find in the literature about that specifically, but it's something that I could
Do you
investigate.
think there might be any other source of bat smell other than poop, which would perhaps be more attractive?
Perhaps. I'm very new in this field. But yeah, maybe. Honestly, when I first, as I said at the beginning of the talk here, I considered the poop to be a neutral scent, which I could have just left the mazes empty, I suppose. But I considered the fact that there being a scent would also maybe be a better source of a control. But maybe if I would do this again, I would use guano, a different conspecific hue, and a control trial, and just kind of compare everything.
Do you know, would the snake skin shed smell the same way as seafood? Or would it have like a less of a smell in the shed?
Yeah, I tried to keep as much of the scent on the shed as I could. It was transported from a little while away in a plastic bag, so we might have lost some of that. What I was nervous about was the shed not being considered a predatory cue, and just more of a general there's a snake that was over here. But I still think it would be related to the snake. I don't see why it wouldn't be. Sir?
I was just thinking, maybe the radial legs is maybe just not quite large enough, and it's possible that maybe the scents all kind of just mix.
Yeah, so the point is that the maze might be a little too small for the scents to really isolate in the arms. And I do think that's a factor. What I did to try and counteract this is in each of the arms of the radio maze at the very end, I drilled a hole to kind of get fresh air pumping through and maybe limiting the airflow outward and more flow inward. But there's only so much I can do about that. But I was very gracious to accept the mazes from another lab who was previously doing locust research, and it worked out great. Thank you.
Okay, next up, we have Beatriz Noguera, who, from the University of Waterloo, who will speak to us on about her project characterizing roost selection of migrating slow regard bats during fall stop.